Okay, this is a second submission for week 6 as a review question. I just have some main points here that are from the question to help me track as I go along. So week 6 was looking at the overexpression of P53 cells in 293T cancer cells. And the first part of the question examines qPCR and what reagents might be needed within this assay. We would need a primer, a nucleotides plus polymerase. We would need a reverse transcriptase, cDNA, and then some form of a fluorescent dye. So our primer would begin this process, the process of splitting the DNA. Then the nucleotides and the polymerase would, through the use of reverse transcriptase, create cDNA, which would be coming from our mRNA. And then our fluorescent dye would help us visually determine the amount of protein present when we did this test. What we might expect to see is that we might have two tables here, both with a fluorescent dye in it. One would be our control, and one would be our upregulation of P53. So in our upregulation of P53, we would hope to see larger fluorescent dye active, directly linking to an overexpression of P53, and as a result, an overexpression or a more present dye. And these would be from the test tubes that we would have done earlier, and we would expect to see less dye in the control, and a lot more activity anyways in the P53 overexpression, which would then translate over to a final results in this graph. The second part of this examines Western blot, which could be used to determine the levels of the protein. And we might have a we might have a table with 3 wells in it. The first one would be our DNA ladder. We would have one for our transfected, namely our overexpression in P53, and then a control. But in order for us to visually see this, we would need a couple of antibodies. So in this case, our P53 is our antigen, and our primary antibody would be mouse anti-human, P53. Our secondary antibody would be goat anti-mouse, P53. And how this would work is that our primary antibody would bind directly to the proteins, but because our horseradish dye could only bind to the secondary antibody, we use 2 antibodies, and this will bind to the first, and as a result, we have an increased concentration of horseradish dye, allowing us to see it much more visually. So our DNA ladder, we already know what these numbers might be. Arbitrarily, we could put 10, 20, 30, 40, and this will just let us visually see our results. This will end up going in a well, and there'll be a current running through it. And as I mentioned before, our primary would bind like this, and then our secondaries would bind like this, and this increased concentration would allow us visually to see it. So what we might expect to see here, is that in our control, we might have a very normal level splotch or blot from the Western blot analysis, and in our transfected one, we might expect to see a larger and much darker band. And our control, we would hope it stays the same throughout the entire experiment. And if we didn't have overexpression of P53, we would expect this to be far lower as well.